So shall we start the session, sir?
Okay, can we start? Oh, I'm
Sir,
ready.
I'll start with the welcome address, sir.
Okay, sure.
Yes, sir. Good evening to all respected dignitaries, eminent speaker, faculty members, researcher, participants, and dear students. It gives me immense pleasure to welcome you all to the 7 days multidisciplinary faculty development program on interdisciplinary AI-driven innovation and digital transformation across disciplines, organized by Indian Society of Research and Development Professionals Association with SKRGC Academy. Over the past 6 days, this FDP has successfully brought together experts from various disciplines who have shared valuable insights on emerging technologies, artificial intelligence, digital transformation, and interdisciplinary research. Each session has inspired us to think innovatively and apply technology for academic and societal advancement. Today, we are privileged to have Dr. T. Sivaraman sir, Professor, Department of Chemistry, VSB Engineering College, Karur, as our distinguished speaker for day 7. Sir, we are extend our heartfelt welcome and gratitude for accepting our invitation and sparing your valuable time to share our expertise with us. I also warmly welcome all the participants who have actively engaged throughout this FDP session. I extend my sincere appreciation to the organizing committee also. I hope today's session will be equally enriching your knowledge. Thank you all. Once again, I welcome you all. Welcome, sir.
Do you hear me?
Yes, sir. Yes, sir, you are audible, sir.
Audible, right?
Yes, sir, you are audible, sir.
Okay. So good evening to all of you. I am Dr. Sivaraman, Professor, Department of Chemistry, VSB Engineering College, Karur. First of all, I would like to express my thanks to the Indian Society of Research and Development Professionals and the organizer behind the event for giving this opportunity to share some of my research work with all of you today. Today, we will be discussing about computational tools for molecular modeling and drug design. For sake of clarity, I have divided the presentation into 3 parts. The first part, we will be discussing about the basics of bioinformatics and drug design. The second part, I will discuss the molecular modeling with a typical example. In the third part, I will come up with an interesting drug designing story using molecular docking studies. So before starting the discussion, just 1 or 2 slides, I'll be talking about my academic path. I trust that the information will be very useful to share effective discussion among us today. So as you can see in the slide, I'm a chemist. Then I became a biochemist, then bioinformatist. So my career starts from MSc to PhD to postdoctoral positions, as you can see in the slide, in various countries. Then I started my teaching and research as a lecturer, then to associate professor, and various academic cadres, as you can see in the slide, in 5 different organizations. Right now, I'm working as a professor in VSB Engineering College, Karur. So in this slide, I'm showing my research interests and as well as the metrics of my research. Simply, my research interest is focused on computational drug designing. So in this slide, I'm showing the milestones achieved so far from my scientific reputations. You can see the first one, HV exchange of proteins. So our lab has demonstrated first time the experimental methods by which the two-state models that are being used to explain the hydrogen deuterium exchange in proteins. And the second one is TCA-induced protein precipitation. As most of you know that TCA is a popular protein precipitating agent. So our lab has given a molecular mechanism by which TCA-induced protein precipitations. Also, our lab contributed to folding pathways of beta sheet proteins. We have developed a number of computational tools, and all the tools are available in the open source and accessible. These are the computational tools which are available and accessible to everyone. And our lab also has designed a number of anti-cancer compounds, anti-venom compounds, anti-viral compounds, antibacterial compounds, and so on. Okay, now let us start the discussion. As I mentioned, first we will discuss about some basics of bioinformatics. Is everything clear? Slides are visible, right? Anyone, please? Slides are visible?
Yes, sir. Your slides visible, sir.
Okay. Thank you so much. Let us start with informatics. What do you mean by informatics? The word informatics derived from the French word informatique, and it means that the acquisition, structure, and sharing the information of data. There are a number of branches, agroinformatics, bioinformatics, business informatics, and so on. We are going to discuss about bioinformatics. So what is bioinformatics? Bioinformatics is a branch of science which deals about the biological data using mathematical expression in conjunction with the computer science. So it's a combination of biology, mathematics, and computer science. Okay. Now there are three important components in the bioinformatics. Database, algorithm, and computational tools. In this presentation, we are going to discuss about computational tools. One interesting point, bioinformatist and bioinformatician. What do you mean by bioinformatist? So bioinformatist is an expert who knows how to operate the bioinformatics tools and as well to manipulate the tool to write interface for effective use of the tools. Bioinformatician, on the other hand, bioinformatician is an expert who can know only how to operate the bioinformatics tools. So that is the difference between bioinformatist and bioinformatician. Okay, now drug designing. First of all, what is drug? Drug can be a small chemical molecule or biological molecule having potential to either to prevent or cure the disease or disorder, and as well to enhance the mental and physical welfare. So what drug? The drug can be designed either by pure experimental methods or by pure computational methods or by combinations. Nowadays, drug designing are being carried out by the combination of experimental and computational methods to speed up the process and as well as for effective outputs. Okay. However, the drug designing is a very complex process, time-consuming, and very expensive process. Why we say complex? Because as per the recent survey, suggests that there are 10 to the power of 60 plus compounds. From the 10 to the power of 60 plus compounds, we have to identify the lead compounds. So, what are lead compounds? Lead compounds are the compounds that enter into the preclinical studies. So from that, we have to identify the drug-likeness compounds. Those are the compounds that enter into the clinical studies. From that, we have to identify the drugs. Drugs are the molecules which enter into the market. So it's a very highly complex process. And if you look at the time, for moving a molecule from the laboratory to the market is about 10 years or so. It's a time-consuming procedure. And if you look at the kind of cost, the development cost for a new drug is about $1 billion US. you can convert into Indian rupees. So it's a very expensive process. So it's a very complex, expensive, and time-consuming process, but it's essential. So the drug designing can be, as I mentioned, can be done by computational or experimental. Now we are going to look at computational methods. Either by computational methods or experimental methods, for designing the drug molecule, we need two prerequisite informations. One is target, another one is small molecule. Target, most of the cases, are proteins. So we should know the protein structure. We should have the three-dimensional structure of protein. We should have the three-dimensional structure of small molecules. Then only drug designing can be carried out at atomic level resolutions. So there are two things are important. One is three-dimensional structure of proteins. Another one is three-dimensional structure of chemical molecules. Now, in this slide, I am showing the number of databases available to retrieve the protein structures and small molecular structures. As you can see, the PDB is a popular database from which the three-dimensional structures of the protein can be retrieved. And other databases like PubChem, NCI, Zinc are the databases from which we can retrieve the small molecules. And the molecule structure may be available in different formats. And there are a number of tools by which the structures can be visualized and analyzed. For example, Argus Lab, PyMol, RasMol, Discovery Studio, Schrödinger. Discovery Studio, Schrödinger are commercial tools. Swiss PDB, Swiss ADME are online tools. And ChemSketch, Argus Lab are standalone tool, but they are free. So there are a number of tools by which the structures can be visualized and analyzed. Okay, now how to get the three-dimensional structure? The three-dimensional structures of the proteins can be elucidated by NMR or X-ray. In this slide, I'm showing the NMR structure. In the slide, I'm showing the BBTI protein, for which the three-dimensional structure would be determined for the first time by using NMR method. Kurt Uttrich received the Nobel Prize for his monumental contribution towards the work in chemistry in the year of 2002. And this is the only technique to date, to my knowledge, to determine the structures of the macromolecules in solution. Okay. The another technique is X-ray crystallography techniques. Myoglobin, hemoglobin are the two proteins, as I'm showing in the slide. The two proteins for which structure could be determined by Max Proust and John Kendall. They received the Nobel Prize in chemistry in the year of 1962 and for their contribution towards the work. So these are the two popular techniques by which the three-dimensional structures of the macromolecules can be determined. Nowadays, electron microscope is also being used to determine the structure at very high resolutions. Okay. Now, theoretically, the three-dimensional structures of the protein can be predicted. There are three different methods we have at present: homology modeling, threading, ab initio. Homology modeling works on the principle that similar sequence will have similar structures. Threading works on the principle of molecule having, proteins having similar physicochemical properties will have the similar structures. Ab initio works on the first principles. As you can see in the slide, in 2024, David Baker received the Nobel Prize for his contribution towards the protein structure prediction. And he shared the Nobel Prize with another two scientists, as I mentioned here, Demis and John Jumper, for the contribution towards the computational structure prediction. So now I think we are very clear about how to determine the structures of the protein. It can be determined by three different experimental methods, or it can be determined by three different computational methods by which the three-dimensional structures can be determined. Now, I will be explaining, as I mentioned in the beginning, the molecular modeling with one typical example. So I am going to explain the homology modeling procedures over here. Homology modeling. In the homology modeling, the structures of the protein can be predicted. In order to predict the structure of the protein, we need a template structure. The template sequence and the target sequence should be very similar to each other. So, now the question is how to get the template sequence. We have the target sequence. For which sequence available? Structure not available. Then we want to get the structure for the target protein. Now we are using the homology modeling. In the homology modeling, we need a template structure. So how to get the template structure? So very simple. Just go to the NCBI website. All are available, open sources and accessible. So go to the NCBI site and select the BLAST tool. On the right side, you can see the BLAST tool. There are a number of BLAST tools. You can select the protein BLAST because we want to get the template sequence for the protein target sequence. So go to the NCBI site and select the BLAST. Then the BLAST, once the BLAST tool is pop up, you can paste the target sequence. Then select the database's protein data bank because we need a similar sequence for which three-dimensional structure is available. So we paste the target sequence in the accession bar and then we select the databases, protein data bank in the databases, then we search. Then we'll get the number of hits as you can see in the slide. Generally, the first hit will be the best hit. However, we have to make sure the target sequence and template sequence have the complete sequence coverage. You see that the first hit have 98% of sequence coverage. And in addition to that, we have to see the percentage of identity, percentage of similarity, and the overall total score. Accordingly, we have to select the template. So we have selected the first sequence as the template to predict the structure of our target sequence. Now we understood how to get the template sequence. So then, for using the PDB ID, we can download the three-dimensional structure of the template protein from the protein data bank. Now we have the template sequence, we have the template protein structure. Then, using the multi-line tool, the target sequence and template sequence can be aligned. The aligned sequence can be taken as a alignment file. Now we have the alignment file, we have the atom file. Atom file is nothing but three-dimensional structure of template one. And we have the script file. Using this, we can run a program. The program is modular. Modular is freely available standalone tool. And it can be downloaded and it can be used. See here, the red one is the template structure. The blue is the target structure. The target one before predicting we had only the sequence. Now we have the three-dimensional structure. If the template file, protein file, sequence file, alignment file, and script files are ready, within half an hour or so, we will get the three-dimensional structure of the target protein. Okay. Now we have the structure for our target protein. Okay. I hope if you have any questions, you can ask me in between, no problem. Okay, let me continue. Now, so as I mentioned earlier, in order to predict in order to do the drug designing, we need three-dimensional structures of the macromolecules as well as three-dimensional structure of small molecules. So far we discussed how to get the three-dimensional structure by experimental methods, computational methods. How to get the three-dimensional structure from the database. How to get the three-dimensional structure of small molecule from the database. Okay. Now we know how to get the three-dimensional structure of small molecules and macromolecules. Now we come to the drug designing part. Now, as I mentioned in the beginning itself, the drug designing can be carried out either by merely by experimental methods or computational methods. In this talk, our focus on computational methods. So just to explain, the advantages, the merits of the computational methods in the drug designing procedure. See, case number 1, as I mentioned in the slide, structures of the receptors are not available. Structures of the ligands are available. Without having the structure of the target, we cannot do the drug designing at high level resolution by using experimental methods. So, but can we do the drug designing by computational methods? The answer is yes. If the three-dimensional structures of the target molecules are not available, then we can predict the three-dimensional structure of the target molecule using our computational tools. The structure can be calculated, it can be validated, and it can be simulated at near physiological conditions. The structure can be used for drug designing. Then the binding site of the predicted structure can be mapped out. Then using the high throughput virtual screening, the lead molecules can be identified. Okay, interesting. I hope everyone follow. If you have any clarification needed, please ask me in between. Now consider the case number 2. Structures of the receptors are not available. Structures of the ligands also not available. So without the structure of the target molecules and the ligand molecule, we cannot do the drug designing at high resolution by experimental methods. Now the question is, can we do by computational methods? The answer is yes, we can do. How? The three-dimensional structure of the target molecules can be calculated, validated, and simulated at near physiological condition. The binding site can be predicted. Okay. Then structures of the ligands not available. What we can do? Using the receptor-based pharmacophore designing, the three-dimensional method, the drug lead molecules can be identified. The method I'm stressing is receptor-based pharmacophore designing. Okay. If you have any clarification, definitely I will explain more. Another case, interesting case, consider structures of the receptors not available, structures of the ligand not available, the small molecule database also not available. So can we do the design? Can we do the drug designing? Yes. That is merits of the computational method. So as I mentioned in the previous 2 slides, the three-dimensional structure can be calculated and validated and simulated at near physiological conditions, and it can be prepared for the drug designing purpose. Now we are using the method called de novo designing. Using the de novo designing method, the lead molecules can be identified. One more case. See, structures of the receptors not available. Structures of the receptor cannot be predicted. The past 3 methods I mentioned, structure not available, but we predicted. Now there is the case, structure cannot be predicted. Then how we can do the designing? The method called QSAR. You can see the slide, QSAR, quantitative structure activity relationship. Using the QSAR drug designing strategy, the lead molecules can be identified. Okay. So this shows the kind of unique advantages of the computational methods in the drug designing process. One important thing is we have to keep in mind, whatever we do, the computational methods should be validated by experimental methods. There is no doubt about it. But the process can be speed up and can be effectively identified by computational methods. And just see this slide. Okay, these are the number of drug designing strategies: structure-based drug designing, high-throughput virtual screening, de novo drug designing, ligand-based drug designing, fragment-based drug designing, core hopping, peptidomimetics, peptidodynametics, and there are a number of methods. Using the appropriate methods, the drug designing can be done by computational methods. The peptidodynamic is a method developed in our laboratory and it's already documented in the literature. If you are interested in, you can go and refer it. The peptidodynamics is the method by which a polypeptide can be decoded into small molecules. That is the beauty of the method under dynamic conditions. Okay, now we are going to discuss about a case study of drug designing done by molecular docking methods. So the disease we consider cancer. Just a few 1 minute I will discuss about. So what do you mean by cancer? As we, most of you know, I hope cancer or the cancers are due to the uncontrolled cell proliferation of unwanted cells in the system. Uncontrolled proliferation of unwanted cells in the system. This is because of a number of factors. One of the factor is apoptosis. So apoptosis is a programmed cell death. When in the case of cancer, the apoptosis is impaired. Because of that, the cancer cells cannot be destroyed. So the apoptosis should be initiated in the case of cancer. So now, this is the strategy behind. Now, these are the small molecular ligands. And these ligands are identified as the anti-cancer compounds. Why? Because these compounds are interacting on the, as you can see the slide, I have shown a protein and I have labeled the BH3 group, BH group. Okay. The three ligands are capable of interacting in the BH3 binding group of the protein. The compounds which are interacting in the BH3 binding group may act as the anti-cancer compound because the protein is the anti-apoptotic protein. Anti-apoptotic protein resists the apoptosis, but in the cancer, apoptosis should be induced. The compounds that are interacting in the BH3 binding group are inhibitor of the anti-apoptotic proteins. So, by inhibiting the anti-apoptotic protein, the apoptosis can be initiated. So, the compounds that are capable of interacting in the BH3 binding group can be called, can be denoted as anti-cancer compounds. That's clear. Slides are visible. See, I think, okay, now you see the slide, I'm showing the molecular docking results of the three ligands on the protein structure. The two ligands, I hope everyone can see the BH3I1, the ligand named BH3I1 and sanguinurine are interacting in the BH3 binding group. So definitely the two ligands can act as a anti-cancer compound. Now see the slide, interesting point. Remember the name BH3I1 sanguinurine. The BH3I1 and sanguinurine are occupying the BH3 binding group, but not fully. The BH3I1 occupy a part of the BH3 group and sanguinurine occupy another part of the BH3 group. So based on the structural features of these two compounds, if we could design a ligand which can occupy the full of the BH3 group, then that ligand definitely will be the best ligand to the protein and can act as a excellent anti-cancer compound. So what we did is, using the structural features of the BH3I1 and sanguinurine, we developed a number of library compounds. From that we came up with two compounds. The compounds are, I have mentioned that you can see the bottom of the slide, XJO34C, XJO35B. So look at the structures of the two compounds. Carefully look at, you will find out any difference. Can you find any difference between the two ligands? If you look at very carefully, the two ligands are very similar. The two ligands are structural isomers. In the case of 34C, the ligand on the left side, you can see the methoxy group. The methoxy group is located the second and third position. I can show in the cursor, second and third position. But in this ligand on the right side, the methoxy group present in the third and fourth position, except that the two ligands are same. But interesting point is the ligand in the left one, shown by cursor, you see, that ligand is interacting with the anti-apoptotic protein, and the ligand on the right side is not interacting. Now, using the experimental methods, we have shown that this one ligand is interacting, another ligand is not interacting. Experimental method can tell only this ligand is interacting, this ligand not interacting. If the ligand is interacting, how it's interacting can be derived. But if the ligand is not interacting, experimental method cannot tell why it's not interacting. We are going to bring the answer by computational methods, how the structurally similar two ligands differ in their binding interaction with the protein. So that is the next few minutes discussion. Hope it's very interesting. The two ligands are structurally very similar. One is interacting, another one is not interacting. We cannot come up with the structural features and the structures which are responsible for the interaction, which are responsible for not interaction by experimental methods. But we are going to come up with a computational method. Let's see how. See, these are the experiments. I think it may get bored for you. Because I want to make you, I want to convince the audience. Yes, one ligand is interacting, another ligand is not interacting. For that purpose only. If it is very hard, just ignore it. But just listen why I'm telling one ligand is interacting and another is not interacting through the experimental evidence. This is called HSQC spectrum, NMR HSQC spectrum. Here I have overlapped the HSQC spectrum of the protein BCL-XL and the protein treated with the ligand. And the HSQC spectrum shows the backbone NH protons. Each cross peak represents the NH protons, amide protons of the protein. The red one are the amide protons of the free protein. The blue are the cross peaks representing the amide protons of the protein treated with the ligand. You can see there is a chemical shift perturbation, and the perturbation is very systematic and only subset. So I can show this in the cursor. I can see red, blue, chemical shift perturbation here and here. You can see here, see here, and it's a very systematic and subset. Mean to say that the ligand is specifically interacting with the protein. So from the experimental studies at a high resolution, we are very clear the ligand is interacting. This is another ligand, the one which is not interacting. We have done the experiment at different stoichiometric ratio of the protein ligand, but not like shown in the previous slide. You can see there is no systematic chemical shift perturbation, and there is no subset of amide protons, and it's totally clumped, meaning to say that the ligand is not specifically interacting with the protein. That's the point I want to make. So experimentally very clear, one is interacting, one is not interacting. Three methods. The second method, see, another method is called saturation transfer double difference method. This method is very powerful to show which part of the ligand is interacting with the protein. So just you need not worry about much here. And these two peaks, as I am showing this cursor, these two peaks are these two peaks represent the methoxic groups of the ligand, and this peak represent the aromatic ring of the ligand. And this is 1D NMR spectrum of the ligand. And here is a STDD, saturation transfer double difference NMR spectrum. When in the experiment, very simple logic is, when we have the peaks in the STD experiment, that corresponding moiety have strong interaction with the target protein. See, we have the strong peak over here. That means this aromatic groups is interacting with the target protein, and we have two methoxic groups. The peak for the two methoxic group. So the two methoxy groups are strongly interacting with the target. So this ligand is interacting. Now you see the ligand which is not interacting is the 1D NMR spectrum of the ligand, and you can see the STD spectrum, very weak signal. Meaning to say that the ligand is not interacting or very weak, have very weak interaction. So when we say weak interaction, what is the weak? We should know, right? So for that, we did a ITC experiment. This is another final experiment I am discussing in this presentation. Because I know that most of you may not be very familiar with biological, biophysical techniques, but this, no, I have discussed this. Unless until you understand, you won't appreciate the computational, the advantages to explain the bustle we have. So this is ITC experiment. ITC experiment can be used to determine the binding constant of the ligand interacting with the protein. So in the top panel, right, in the each spike, we are getting upon adding the ligand to the protein, and the area under the peak can be integrated, and we have the data over here in the bottom panel, and the data can be fitted to an appropriate equations to yield the binding constant. So the binding constant about 3.4 micromolar is very appreciable. When the binding constant is about micromolar or sub-micromolar, means that is very appreciable. So we have about 3 micromolar binding constant. See the another ligand, we have the binding constant about 750 micromolar, meaning to say that the ligand is not interacting. When the binding constant is more than 500 micromolar, it is not appreciable. And because of any type of interactions, we can have some values. So the benchmark is when the binding constant is 500 or above 500 micromolar, the ligand is not specifically interacting. 10 micromolar or less than sub-micromolar is highly preferable. Nanomolar is really preferable. Okay, so now from these experiments, very, very clear that we have taken two structurally similar ligands. One is interacting, another one is not interacting. Now, question is how structurally similar ligands have differential interaction with the same protein. We are going to address by computational method. Okay. Now we come to the final part of the discussion today. So we have taken the ligand and we did the molecular docking studies. And the ligand perfectly fit into the BHD binding groove of the protein. You can see the ligand is perfectly docked in the BHD binding groove of the protein and well converged with the binding energies. Meaning to say that this ligand is very specific to interact with the protein. Okay. Now, the ligand which is not interacting, we did the molecular docking. You can see the ligand is perfectly fit into the BHD binding groove of the protein. But however, the energy not converged. So that means it has some kind of interaction, but specifically it shows the interaction on the BHD binding groove only. Again, perplexing. So now what I did is I have overlapped the molecular structures of the two ligands on the BHD binding groove of the protein. The ligand shown in magenta is the ligand which is interacting. The ligand shown in the cyan color is the one which is not interacting. But you see the beautiful overlap between the two ligands, and look at this very carefully. And these are the, I am showing in the cursor, these are the methoxy group, 3 methoxy group. In the magenta, it is in the second and third position. In the cyan, is the third and the fourth position. Okay. Now, I have done the electrostatic mapping of the BHD binding groove of the protein. And in the left side, I'm showing the front side view of the binding of the ligand on the protein. In the right side, I'm showing the binding of the ligand to the protein from the rear side of the BHD binding groove. So what you can see the methoxy group, one of the methoxy groups of the ligand not binding is exposed to the charge incompatible environment. Charge incompatible environment. The methoxy group, OCH3 group, is exposed to the negative charged environment, is charge incompatible. Now, in order to bring the methoxy group into the group, the ligand need to adapt a conformation. Now look at one simple chemistry. See, the two ligand, in the ligand, two 6-membered ring connected by a single bond. When the two 6-membered ring connected by a single bond, the two ring will appear perpendicular to each other. That is the lowest energy, thermodynamic lowest energy. So the two rings will be perpendicular to each other. You see the diagram I'm showing in the slide. Very beautiful. This is one 6-membered ring. This is another 6-membered ring. They are perpendicular to each other. Because the two rings are connected by a single bond. Now, in order to bring the methoxy group into the BH3 group, I'm showing, see the cursor, the 6-membered ring need to adapt a conformation, which will affect the 6-membered ring of this part. Accordingly, the rest of the part will be affected. So what I like to say, in order to bring the methoxy group into the group, the ligand need to adapt a conformation. The resultant conformation is not compatible. To fit into the group. So the problem, the answer is the presence of the methoxy group. The presence of the methoxy group at the 4th position is crucial for the ligand not to interact, because that methoxy group is exposed to the charge incompatible environment. Now you can see, very beautiful. So this is a protein and the BH3 group and the two ligands are interacting, but in one of the ligand, the methoxy groups is exposed to the charge incompatible environment. Now you can ask, so what? See, the results have been published in the journal, as I have shown in the slide, published in the Journal of Medicinal Chemistry. Now, the important outcome is, using the 4th positions, we can design a very specific ligands for the different anti-apoptotic proteins. Actually, we have, to date, we have about 6 different anti-apoptotic proteins. Each anti-apoptotic protein play an important role in different cancers. We have 100+ cancers. So we can, and each cancer overexpress some particular anti-apoptotic proteins. Some cancer overexpress all type of anti-cancer compounds. So using these structural features, we can design a very specific anti-cancer compounds. The structural features would be derived from the computational method. Now, using the methoxy group and the positions, we can develop a number of ligands which may have a very specific interaction with the towards the anti-apoptotic protein. So I bring the conclusion here. So what I have to say from here, computational tools can be successfully used to screen lead compounds and as well designing de novo small chemical molecules. The in silico tools are indispensable to efficiently speed up the drug designing process in cost-effective modes within a stipulated time frame. Very important point is, the last but one, you can see the structurally similar ligands may not exhibit structural binding affinities to a receptor protein. So during the discussion, I have skipped many slides because I thought it may not be appropriate, and because time constraints, and I didn't discuss those informations. For those slides also, I have brought the conclusions in the slide. So whatever we discussed at present moment, this is the important conclusions. So the take-home message is, similar ligands need not to have the similar interactions. computational methods play an important role in to speed up the process and as well to effectively identify the drug molecules. So that is the message from these presentations. If you have any questions, please feel free to ask me. And here is you can ask me now. I will try to answer. If you have the questions, you can contact me over email also. I have given the email address in the slide. These are my social activities. I have my own YouTube and blogs. If when you have leisure times, you may feel free to go and explore those informations. You may like it, I hope. And so thank you so much for your attentions. Any questions, please ask me. Thank you. Vetrivel, are you here? Vetrivel? Okay, any questions, please? Vetrivel, do you hear me?
Sir, yes, sir. You are audible, sir.
Yeah. So I completed the talk. Any questions? It's a question and answer session. If any questions, I will answer.
Okay, okay, sir. Participants, if you have any queries, you may ask now.
Okay. So no questions, I think.
Sir, I think yes, sir. No queries, sir. Shall we
Okay.
conclude the session, sir?
Yeah, sure, sure.
I will conclude the vote of thanks, sir.
Okay.
Good evening, everyone. As we come to the conclusion of this wonderful 7 days faculty development program on interdisciplinary AI driven innovation and digital transformation across disciplines, it is my proud privilege to propose the vote of thanks. First and foremost, I express my sincere gratitude to our esteemed speaker, Dr. T. Sivaraman sir, Professor, Department of Chemistry, VSB Engineering College, Karur, for delivering an insightful and thought-provoking session today. Your knowledge and experience have greatly benefited all the participants, sir. I extend heartfelt thanks to the Indian Society of Research and Development Professionals, ISRDP, and SKRGC Academy for organizing this excellent FDP and creating a platform for interdisciplinary learning and professional growth. My special appreciation goes to the resource person from day 1 to day 7 for sharing their expertise and enriching us with a diverse perspective on AI driven innovation and digital transformation. I would also like to thank the management organizing committee, coordinators, technical support team, and volunteers for their dedicated efforts, planning, and seamless execution of this program. A special note of gratitude to all the participants from the different institutions and disciplines. Your active involvement, interaction, and enthusiasm made this FDP highly successful throughout the week. Finally, I thank everyone who contributed directly or indirectly to this success of this event. Let us carry forward the knowledge and inspiration gained through this FDP into a teaching, research, and professional practices. I I'm willing to thank you all. Thank you, sir.
Okay, thank you.